12thBio-botany, Chapter 4, Principles and Process of Biotechnology. In this part, we are going to discuss about bioassay of target gene. A bioassay is an analytical method. Bioassay is a broader term. It covers the analysis of various biomolecules. But here what we are going to discuss is the bioassay for the target gene effect. What is target gene? Target gene often just means gene of interest or a particular gene being studied or manipulated in an experiment. We may use a gene in an experiment, we may modify a gene in an experiment. We will like to see the effect of the gene. That gene is target gene. In recombinant DNA technology, a foreign gene is inserted into the host. The foreign gene will produce a product. If we are interested about the product, that is gene of interest, we will like to see the effect of that gene. That gene is called as target gene. We may not know the function of particular gene, but we will like to study that gene. That gene is target gene. Gene targeting is the study of target gene effects. If we want to study the effect of target gene, we need a technique. The effect of target gene is studied by some techniques. Target gene's effect is studied by homologous recombination. Before we understand what is homologous recombination, let us see what type of changes are made by homologous recombination on target gene. The homologous recombination modifies the target gene. Homologous recombination modifies an endogenous gene. Endogenous gene is the gene which is inside the cell. Homologous recombination can be used to delete a gene, add a gene, and introduce point mutations. Homologous recombination modifies individual base pairs and causes mutation. So the function of gene will be stopped. Homologous recombination deletes a gene. When a gene is deleted, its product will not be formed or a particular character will not appear. From that we can understand the effect of that target gene. Homologous recombination adds a gene. When a gene is added, a new character appears on the organism or a new product will be formed, so that we can understand the effect of the newly added gene. Homologous recombinations is a type of genetic recombination in which nucleotide sequences are exchanged between two similar molecules of DNA. This is the DNA sequence of Plasmodium. This is the DNA sequence of another organism. Here some regions are homology regions, similar nucleotide sequences. In between the two regions, the dissimilar nucleotide sequence is there, but this homology regions involve in exchange of DNA. That is called as homologous recombination. Here there are two DNAs. There are certain areas the DNAs have homology. In the homology regions, recombination takes place. So a part of the gene is removed and another part is added, so that the gene will be made inoperative. The functioning of gene will be stopped. This is gene knockout. A gene knockout is a genetic technique in which one of an organism's genes is made inoperative, knocked out of the organism. The gene may be removed or gene may be inside, but it cannot function. That is gene knockout. This is done to study gene function, usually by analyzing the effects of gene loss. Gene knockout results in gene loss. The functioning of gene is lost. So we can understand the gene function. We can study the gene function. For that we need vectors. Vectors do this work of gene knockout. So here this DNA sequence is target gene sequence. Another one is vector. Vectors involved in this homologous recombination. Gene targeting requires the creation of a specific vector for each gene of interest. The vector must have homologous sequence with the gene of interest. So for a particular target gene, a specific vector is designed. For this purpose, two types of targeting vectors are used. One is insertion vectors. Insertion vectors are completely inserted. Replacement vectors replace a part of the gene. Insertion vectors are completely inserted into the target gene. Replacement vectors replace a part of target gene. So this is insertion vector, it is completely inserted. This is replacement vector, it replaces a particular sequence. This is target gene. Insertion vectors are entirely inserted into the target locus. This is an insertion vector, it is circular in the beginning. The insertion vectors have single restriction site. At one place restriction enzymes cut on the vector as well as the target gene. So the ends of vector is ligated, fused with the target gene. After insertion only it will be linearized. Initially these vectors are circular. The circular vector is inserted on the single restriction site. The vectors are linearized within the homology region. Within the homology region the vector is inserted and it will be linearized. So here the vector is linearized. Already the vector sequence matches some other sequence. So there will be duplication of the sequences. It leads to duplication of sequences. The vector is linearized within the homology region. So some regions are repeated duplication. This duplication stops the functioning of gene. So we can understand its effect. This is target gene. This is vector. And this is the homology region between target gene and vector. The vectors are linearized before insertion in the case of replacement vectors. The vector is linearized prior to transfection. Artificial transfer of foreign gene is called as transfection. The replacement vector has a homology region and it is collinear with the target. This is a homology region. It collinears with the target. A part of the target gene is collinearized with the vector. And these two parts are the two nucleotide sequences are exchanged by homologous recombination. Then a crossing over occurs to replace the endogenous gene with incoming gene. This is endogenous gene inside the cell. This is incoming gene. This is replaced by incoming gene by exchanging or crossing over. This crossing over results in exchange of the gene. Here genes are exchanged between the homology region of vector and target gene. So the gene will be inoperative. The effect of target gene is assessed after its replacement. So in this part we have understand what is bioassay of target gene, what is target gene, and what are the two types of vectors which are used to understand or study the effect of target gene. In the next part, we will meet with another topic, that is gene editing.